Okay, welcome to slice 3 of these ECG preparatory materials. So far, just to recap on what we've done, we've gone through the order of the chambers in where blood is moved through the heart. Then we went through a fairly in-depth but relatively quick version of the cardiac cycle. The job now is to get into a little bit of the almost like the microanatomy of the heart, and then to talk about, well, what happens and how things can go wrong, before we then move into sort of really getting into the electrics of how everything functions. So what I wanted to do was just do a little bit of a recap, and what you can see, I've just sketched out a very rudimentary version of a sarcomere over here. And we'll just have a little recap of exactly what's going on. So what we've got here is we've got a thick filament, which is called myosin. We've got our thin filament, which is actin. And then we've got M line, Z line, and we need to remind ourselves of how it is that when we get the interaction between actin and myosin, that these Z lines move closer towards the M line. Right, so what we've got with this, if we want to really kind of set it up so that we can figure out exactly what's going on, we need to kind of just take a bit of this, bring it over here, and then make everything a little bit larger. What we've got with actin is, for all intents and purposes, we can think about actin as being like strings of pearls that are wrapping around each other in a helical fashion. Okay, so if we just sort of colour these ones in like this, and then what we're going to have wrapped around this is another filament of actin that I'm not going to colour in. Okay, so it kind of works its way down like this, crosses over like this, like that. Okay, so you can see that we've got this interlocking helical kind of configuration going on. Now, thinking about it, I suppose I should have even drawn that in green, so I had blue in my hand and I'm not going to rub this out and do it again. So what we've got in and around this is we've got our thick filament. Okay, what I'm going to do is I'm going to draw our myosin like this, and then I'm going to draw a head of myosin coming out like that. Okay, because what you need to know about myosin is that kind of looks like, I suppose if I was to just draw a myosin filament like this, what we'd have is you'd have cross bridges like this, and they kind of stick out all the way around, and it kind of looks like golf clubs that are being sort of twisted around each other. Let's just get rid of that for a second. So why am I talking about this stuff that you kind of probably already know? Well, there is important bits going on. So what we've, back to blue, what we've got is each of these individual molecules of actin, okay, each of these individual molecules of actin can sort of best be thought of as globules, okay? And the reason I say that they can best be thought of as globules is because, well, we call them G-actin monomers. So each of those individual pearls of the string of pearl is a G-actin monomer, and then what we've got then is we've got the fibre that it makes up is F-actin. Okay, F-actin, like that. And then with myosin, we've got head and tail, like that. Okay? I'm building to something, I promise you. What we've then got running throughout this is a molecule called tropomyosin. Okay? We've got tropomyosin that runs through it. Attached to the tropomyosin, we've got a particular molecule that is going to become very, very, very important to us. Okay? It's got 3 parts, like this. Okay? Like that. This is troponin. And what the troponin will do, if you just pick it up and kind of drop it in here, okay, we've got a little bit like that. Okay, that is a calcium binding site. Then we've got one of these, which is attached to a G-actin. And then we've got another one, which is attached to the tropomyosin. Now, I'm a big fan of keeping things relatively straightforward. Okay, it's kind of like the way I do things. I always say that I'm a physiologist of very little brain. And me keeping this straightforward is by kind of like having rules. So I always say the Lewis's rules of stuff binding in science. One is stuff binds to stuff. Okay? And when stuff binds to stuff, it changes stuff's shape. And then that affects the ability of stuff to bind to stuff. So what happens with this is, let's just draw in a molecule of calcium. The molecule of calcium will attach to its binding site, and for all intents and purposes, we could redraw this, I suppose, just to illustrate the point slightly better. And what we could do is we could say that there's a little hand here, and then that binds like that. And when the calcium binds in, troponin goes like that. So basically, it goes from this back to the way that I originally drew it, like that. And what that does is it yanks up the tropomyosin away from what is underneath this little bit of the actin filament. And that is a binding site for myosin. Well, why is this important, Lewis? Well, the reason that it's important is because of calcium. What this tells us, because in the heart, obviously we've got no muscular integration whatsoever to be found. So if we want to increase, write this down here in green. So if we want to increase the contractile force, try that again. If we want to increase the contractile force of our heart, the only way that we can do it is by increasing calcium. So if we increase what's called contractility, okay, increasing contractility is done by increasing intracellular concentration of calcium. And as long as we can remember that, then in terms of cardiac function, everything becomes a little bit more straightforward. And we're going to pick this up in the next slice, which is going to be about how we regulate that.